Well, good afternoon, everyone. I welcome you to our webinar series titled MicrobeTalks Exploring the Microbial World, which is hosted by the Puerto Rico Society of Microbiologists. So today we have, well, first of all, my name is Natalie Melendez. I am the social media chair of the Puerto Rico Society of Microbiologists, and I will be today's moderator for this session. So now I will begin introducing our speaker, Brayan Vilanova Cuevas. He earned his bachelor's degree in microbiology from the Inter-American University of Puerto Rico Metropolitan Campus, graduating summa cum laude. During his undergraduate studies, Brayan was a junior research assistant with the Puerto Rico Imber Fellowship under the guidance of Dr. Filipa Godoy Vitorino at the Microbial Ecology and Genomics Lab at the University of Puerto Rico Medical Sciences campus. Brayan has a strong research background with experience ranging from marine microbiology to the gut-brain axis, including significant work at the University of California at Davis, where he completed his prep training at the Guru Lab, studying the effects of antibiotics, infection, and disease on the gut and brain function. Vilanova is currently a microbiology PhD candidate at Cornell University, currently working in the Houston lab. There, Brayan focuses on elucidating the mechanisms of pathogenesis and transmission of the disease Scutociliatosis, which affects the sea urchin diadema, while also studying the immune responses against the disease. Brayan has received numerous awards and honors throughout his academic career, such as the National Science Foundation Graduate Research Fellowship and the prestigious College of Agriculture and Life Sciences Statham Fellowship from Cornell. He has done oral and poster presentations of his research in various national and international conferences, including the 2024 Ocean Sciences Meeting and the Association for the Sciences of Limnology and Oceanography. His leadership roles include being a member of the executive board of different student associations and founding the Latino Educators Advancing Research and Knowledge Crew, an initiative that started after 3 years of doing outreach to middle and high school students from Puerto Rico schools, reflecting his commitment to outreach and education. So without further ado, I'll leave you guys with Brayan.
Thank you so much, Natalie. Appreciate the warm welcome. Hi, everybody. I'm going to just share my screen really quickly here and we can get started. So first off, thank you so much for having me today. I'm really appreciative of the opportunity to talk with you and to share a little bit about my journey while doing my PhD here at Cornell. So today's talk is titled Diadema enterarum's Microbial Community Changes Before and After Mass Mortality Events and How It Compares to Mesocosm Experiments. And so today I'm going to talk about a lot of different aspects of Diadema entellarum, but I do want to be clear that Diadema entellarum is not the only urchin that has been affected by the disease. Most recently published Diadema setosum and other species in Diadematidae family have been affected by this disease. And so before I get to it, a little bit about my journey, and I won't take too long here just because Natalie gave a great review of it. I come from Puerto Rico, as most of us do, and I was at the Escuela Superior in the Inter-American University for my high school diploma, and then I did my bachelor's degree at the Inter-American University for microbiology. I did an RU program with the University of Pittsburgh where I looked at antibiotic resistance and how it affects gene regulation. And I worked primarily with Dr. Filipa Godoy, and these are some of the works that I worked with her on, and I am super happy to be joining you today because I come from this great family of people residing in Puerto Rico doing research. I worked with Dr. Melanie Garreau at the University of California in Davis, and I worked on the human gut-brain axis, and we worked with mice models. Some of my work also focused on Angelman syndrome, which is laughing baby syndrome. and we looked at how the gut microbiome can affect different aspects of the disease. And now I work at Cornell in the middle of upstate New York in Ithaca. And this is my PI, Dr. Ian Hewson, and I work mostly on pseudococceliotosis, which is a disease caused by pseudococceliotes. And ciliates are protists that are single-celled protists that can cause many beneficial things, but also can cause disease. And we'll talk a little bit about that later. Oh, there we go. So why do we care? What are echinoderms? Echinoderms serve as predators and prey in the ocean food web. And so here we have Echinodea, Sideroidea, holothurians, asteroides, all of these different families of organisms that live in the seafloor help with maintaining ecological balances by allowing for coral settlement and maintaining our coral reefs. Also, some of the species like the sea cucumber burrow into the soil and allow for nutrient cycling by oxygenating the soil, which is very important. The one we're going to be focusing on today, though, is the sea urchin, and specifically Diadymetidae family sea urchins. And they are very important because they maintain the coral algae balance. Without sea urchins, we get more algae and less coral settlement. And so why is this work important? Diadema antillarum has experienced 2 mass mortalities to record so far. And the first one was in the 1980s, 1983 to 1984, where 99% of Diadema antillarum died of an unknown disease. After that initial mass mortality event, only 3% of the disease of the animals that were in that environment recovered all the way to 2022. So let's get the bigger picture on that. 99% of that community was diseased and died, and only 3% of that initial community was able to repopulate in the area before another mass mortality event occurred. Now with the mass mortality event that occurred in 2022, we were able to discover that the cause of disease was Diadema pseudococceliotosis. And the cause of that disease is Diadema antillarum pseudococceliotosis phylaster clade. And we'll talk a little bit about what I mean by phylaster clade. Phylasterides are a family of ciliates, and a lot of different ciliates within that family are associated with disease within the marine environment. So why do we care about ciliates? So now that we know why we care about urchins, why do we care about ciliates and why are they important? So just like with bacteria and with other organisms, we never want to see something as strictly pathogenic. So ciliates are not strictly pathogenic. They have a great role in the microbial loop as they bring back dissolved organic matter, and they also feed the gracer chain. So they feed zooplankton and phytoplankton and fish. And so ciliates can consume bacteria, they can consume tissue. When I say they can consume bacteria, I will refer to them as bacteriivores. When I say they can consume tissues when they're affecting other organisms, then they are histophagous. And so they are really important in the marine environment because they allow for nutrient cycling. So what do we know about Diadema pseudococceliotosis, and specifically the ciliate that causes Diadema pseudococceliotosis? Well, we know that this ciliate can cause disease 4 to 6 days after the initial infection. It can trigger immune responses from the urchin, and it burrows into the tissues of the urchin. And as we can see here in image E and image F, those are stains of the ciliate within the urchin's tissues. The image above that is the image of the ciliate itself, and you can see it very easily under light microscopy. We have now found that this ciliate is both bacteriovorous and histophagous. So what we mean by that is you can culture the ciliate in a falcon tube with 15 mils of water and yeast extract for bacterial growth, and it'll grow fine. Histophagous, on the other hand, by that we mean it can consume the tissues of Diadema antillarum and other urchins and cause disease and mortality. This ciliate is closely associated with another ciliate called Phylaster digitiformis, where the name comes from when we say phylaster clade. And so here on the right side of the screen, you can see the phylogenetic trees where our ciliate falls into relation with other ciliates nearby. And they all belong to the Oligohymenopharia group, or Scuticociliata. And there are plenty of marine diseases caused by ciliates within this family, which is why we find this to be a really important thing to study. We need to understand how these ciliates cause disease, but also how can we manage those diseases. And so for today's talks, I want to address two questions. How does Diadema scuticociliatosis affect Diadema entelarum's microbiome in the environment and in experimental setups? And the goal with the experimental setups is to understand that when we replicate a disease in the experiment, in the lab, are we getting the same microbial interactions as when we are in the environment? And the next question we will be talking about a little bit later in the presentation, but it goes, can we identify similar shifts in the microbiome of Diadema entellarum on historic ethanol preserved specimens before and after the 1980s mass mortality event? Is this ciliate present? And so to start, let's talk about the methods and the data analysis very briefly. I don't want to use too much of our time on this, but we collected samples of Diadema entellarum, and I apologize, the graph seems to be a little distorted here. We collected samples of Diadema entellarum in the environment. We then extracted DNA from those samples, and we focused on 5 tissue types: the body wall, the spines, the coelomic fluid, the gut, and, and the intestine and the gonads. Sorry, this is moving the presentation a little bit weird. There we go. Okay, so we collected from 6 jurisdictions in the Caribbean, and we have 3 recognized health status. So there is reference urchins. By reference, we mean urchins that are in an area where there's no disease in the Caribbean. We have normal at disease site, which refers to urchins that are seemingly healthy, but they are in an area where other urchins have passed away or by the disease. And we have diseased urchins, which show clear signs of the pathogen and the disease. And the data was analyzed to explore differences between health status. I wanted to pinpoint that today in this talk, you will be seeing a lot of 3 different types of graphs. And I wanted to mention, when we look at a taxonomic plot, we are most likely looking at relative abundance of a percentage above. So in this case, 5% or more of the bacteria that are there. The goal of those graphs is to look at the overall microbial community and major components of it. We're not looking at smaller taxa or less abundant taxa. In the core bacteria, when we say core bacteria, we mean bacteria that are at a 90% detection rate in 90% of the samples within a subgroup, and then 0.75 prevalence. So they have at least that much in abundance within that sample group. And then when we look at alpha and beta diversity, but in this case, alpha diversity, we're looking at SHA-1, which is an index for bacterial richness, and Shannon, which is an index for bacterial diversity. And so here's the first example of those graphs. And what we're looking at is a separation between body, the sample type, which we have body wall, coelomic fluid, gonads, intestines, and spines. And we are comparing the richness in panel A and the diversity in panel B. And what we see is that we have a significant decrease of richness of the bacterial community in samples from reference to abnormal. So from healthy subjects that have never been exposed to the disease to unhealthy subjects that have the disease. We have a significant loss of richness in the body wall, the gonads, and the intestines, but it doesn't seem to affect the diversity of the microbes. And what that tells me is that we might have slight changes in the microbial composition, but there won't be significant changes to the overall picture of it. For beta diversity, what I can see here is that there is, what we see with the statistical analysis is that there's no significant differences based on jurisdiction or health status. And this is also separated by body type here, by sample type. There's no significant dissimilarity, which tells me that there's no grouping based on where the sample was collected from or the health status of it. Here, this image will be difficult to see, so I will amplify it a little bit, but I want to focus on specifically what is the taxonomic composition and how it differs with disease. And so something I want you to focus on is this is body wall, coelomic fluid, gonads, intestines, and spines. And when we looked at differences, significant differences in richness, we saw those in the body wall, in the gonads, and the intestines. And I want to focus on that blue color right there. And that blue color seems to increase when you go from reference, which is in the left, to abnormal, which is in the right. And that group is called Fungia hongkonensis. And Fungia hongkonensis is a bacterium whose family is associated with celiac diseases, and being endosymbiotic in cases or symbiotic in other cases to celiac diseases, and allowing for the progression of the disease. But we haven't tested that with this disease in particular, so that's not the focus of this talk. It's more to pinpoint that this bacteria is increasing with the abnormal. The other color I want to point out is the pink in the bottom, and that is Vibrios. And Vibrios increase with disease in marine environments because they like to group where there is tissue loss and detritus. And so Vibrios increase as a sign of that there is disease there. And so how are they significantly enriched? How are these bacteria specifically significantly enriched? And Cateobacter tinospiralis, also known as Fungia hongkonensis, is consistently associated with abnormal urchins. And when I say it's Cateobacter tinospiralis group, is the group where Fungia hongkonensis lies. So what we were able to identify is, sorry, one second. I can see all of the faces on the way and I can't see the slide. There we go. So when we look at the intestines, we see that this Fungia or Cateobacter-like group is in the intestines and in the coelomic fluid. And Propaginium and Microbiome are genera that are consistent with other studies of Diademantularium's microbiome, and they're only in reference or healthy samples. So our goal is to say the Godoy Lab in Puerto Rico has done extensive work on healthy urchins and their microbiomes. And this study shows that it's consistent. Propaginium and Rosemarinus are consistently associated with healthy subjects. While when we look at disease subjects, we see an increased abundance of these Cateobacter or Fungia hongkonensis bacterium, as well as Microbiome. And what we wanted to tell is this a biomarker of disease? And so we looked at what are the core bacteria. So what are the bacteria that are most present and most prevalent in the samples that we're looking at? And so what we see is that Gistimonas, Propinogenium, which are associated with healthy subjects, are abundant in our normal and reference samples, which are healthy subjects. And the only bacteria that's associated that has the detection and prevalence across all samples from the abnormal group is Fungia hongkonensis, which is why we've declared that this bacteria is most likely a biomarker of disease, since it is highly prevalent only in diseased organisms. And let's shift gears a little bit, and let's go back and think, okay, all that data was from the Caribbean, but how does this relate to disease in other areas? So the disease has now spread from the Caribbean to the Mediterranean, the Indian Ocean, the Red Sea, and it has affected other species of urchins. And so we were able to collaborate with a great group of researchers in Reunion Island, which is right there in the map next or near Madagascar. And we were able to look at the microbiome of these deceased urchins. Sadly, we were not able to collect healthy urchins because the population was depleted, but we have samples of diseased urchins. The goal was to look at whether or not the ciliate was present, and then do we find the same bacterial biomarkers? And so when we compared diseased organisms to healthy organisms, what we see is that this Fungia hongkonensis comes back again, and it's the only bacterium that is common between the Caribbean and this samples from the other side of the world showing the same disease. And this was mind-blowing for us because we did not expect this bacteria to have this tight association with disease as it does. So now for the experimental side. while we can see this in the environment, can we cause disease in lab? And so this is from our study from 2023, where we found the disease. We were able to cause disease within 4 days, and we started to see proportion of, this graph here shows the time after inoculation with the ciliate and the proportion of grossly normal urchins. And so what we're looking at here in the gray circles is the proportion of urchins that started losing spines after the disease started occurring. And so we then took these urchins that were diseased, we extracted DNA, we did the same process as the samples before, and we looked at what are the core bacteria in the normal. So how does our normal from the environment and our normal from the experiment compare? And then how does our normal from the environment and our normal from the challenge compare? And what we see is that while Pangia hongkonensis is a biomarker for disease in the environment, it is not as prevalent or as detectable in the abnormal. Not to say that it isn't present, it is, but because of our limited sampling pool, because we only had 4 samples that were abnormal and 3 samples that weren't, it's not as prevalent as when we looked at it in the environment. And we believe this can be based on sample size, but it could also just be that the conditions in the lab are not favorable for this bacterium to occur. So this gives us an idea that while it can be a biomarker for disease, it is not necessary for disease to occur. The other thing we want to note is that we are able to maintain the health-associated bacteria in environment and challenge experiments in the normal urchins, and that's panel A. And so what we're looking at is Propaginium and Kistimonas remain shared in healthy urchins, which allows us to understand that while we are moving the urchin from its environment into experimental conditions, these were lab-reared urchins and they still maintain the communities pretty well. Overall, the bacterial composition in these experiments show that yes, Pangia hongkonensis is there, but it is very minimal in the challenge experiments. Yet it does surpass the 1%. It's higher than 1% relative abundance, which to us tells us it might be a close associate, but it doesn't hold the detection or prevalence threshold to meet the core bacteria. And then Microbiome is not even present. So Microbiome was another one of the bacteria that was associated with disease, but we don't see it here either. So what we're getting at is in mesocosm experiments, we do not find the same biomarker. We see it, but it's not as detectable or prevalent. So in summary, disease causes no big overall shift in the microbial population, but it does shift specific taxa that are associated with disease across disparate geographical locations. And then the experiments in lab-reared animals show that healthy urchins can maintain health-associated bacteria, and with disease, it doesn't seem to show the same biomarkers as in the environment. And so now to answer the second question, can we identify similar shifts in the microbiome of Diadema entellarum in the historic samples, and is the ciliate present? So to detect for the ciliate, we run a PCR to look for this 18S of the ciliate, and we're able to detect it within the historic samples if it's there or not. And then for the 16S amplicon, what we did is we went to the Smithsonian, and there's a picture here while we were there, and collected samples from the Smithsonian's echinoderm collection from 1980 to 1984. And the samples were kept and preserved in ethanol initially, and then we moved them to RNA later and proceed to genomic DNA amplification and microbiome analysis. And if somebody has questions about transport of the ethanol preserved samples, that is why we moved them to RNA later. And so richness, diversity, microbe distribution, how do these samples even look? They are 40 years old. And so we started looking into that, and we wanted to classify it between pre-mass mortality and post-mass mortality. And what we identified is that, again, CHA-1 means bacterial richness and Channon means bacterial diversity. What we saw is we see a decrease in bacterial diversity based on pre and post-mass mortality. Keeping an eye on all of these samples are from healthy urchins because the samples were preserved from healthy urchins, and they were not initially collected specifically for microbiome purposes. So the cautions that we take are not the same. All this data should be taken with a grain of salt, and we're doing further analysis to make sure that we can put a little bit more emphasis on what in the data is true and what could be caused by sample collection. When we look at dissimilarity in the samples, we don't see any grouping based on the time point. So again, we don't think that we have any specific changes in the overall community. The main takeaway from this figure here is that the microbiome has certainly changed in these animals over 40 years' time. And so the main thing is bacteria that are prevalent now here are not the same Quistimonas and Propaginium that we were seeing in the previous part of the study, but we're now seeing Pterosachiella and Neptunibacter that are highly abundant in these ethanol preserved samples. And we were particularly interested in those because they are also bacteria that we see in our cultures of the ciliate. And so this is interesting to us mostly because we believe that these are weedy bacteria that could attract the ciliate to the urchin, but there's no real explanation for that just yet. Right now, what we wanted to show is that there's no significant changes between the groups pre and post mortality, and we don't really see a shift. Again, we didn't see one with the previous samples from 2022, so we weren't really expecting to see a major shift, but we also don't see any biomarkers of disease. And for the biggest question in this part of the study, can we identify the ciliate? Well, we did the PCR and 18S of the gene of the 18S PCR for the samples, and we were able to identify that, yes, based on 18S, we are able to detect a ciliate that is closely related to our ciliate in culture, and that is those samples that you see in blue. And they are from 1981, 1982, 1983, and the rest group a little bit separately, but still within the same branch. And so what that tells us is the ciliate must have been present or a ciliate closely related to it. And we're doing further testing in our future directions are to do multi-loci confirmation. So looking at the 28S, COX-1, and a few other genes to confirm that it's the same ciliate or see what type of ciliate it could be. And then for a summary, we can definitely identify something similar to the ciliate that we have now based on 18S. And there is a significant reduction of microbial diversity, but this could be due to sample size. And there's a lot of other factors that influence that. Before we're done, I'm going to switch gears into Spanish, and I want to talk a little bit about outreach. Y muchas gracias por estar aquí. Ahora voy a cambiar a español. Esta iniciativa es latinos educadores avanzando la ciencia, research y el conocimiento. Es una iniciativa que yo le acabo de dar un nombre ahora este año, pero llevo haciéndola ya varios años, donde le damos charlas a estudiantes de high school y de intermedia sobre lo que sea que estamos estudiando. La idea es exponer a nuestros jóvenes a lo que se está estudiando, cómo se estudia e interesarlos en la ciencia. En las próximas 2 semanas yo voy a estar en Puerto Rico haciendo trabajo con Learn Crew, donde voy a estar impactando alrededor de 1.500 estudiantes, hablando sobre erizos de mar, biología marina, investigación, y ayudando a los estudiantes más avanzados en escuela superior con sus aplicaciones a universidad y hablando un poco sobre eso. Quería proponerlo porque estoy aceptando estudiantes graduados que estén interesados para trabajar conmigo y poder seguir dando estas charlas en el Caribe y en otros sitios en Latinoamérica. Con eso, le doy 1000 gracias a mis colaboradores y a ustedes por invitarme. Y aquí estoy expuesto a preguntas. Ahí tienen algunas fotos generadas con AI sobre erizos de mar, porque en mi laboratorio nos pasamos haciendo muchas fotos con AI. Y nada, muchísimas gracias.
Muchas gracias, Brian, ¿verdad? For such an excellent presentation. Ahora, ¿verdad? Abrimos el piso para todos aquellos que quieran hacer alguna pregunta, ya sea abriendo sus micrófonos o escribiendo por el chat. ¿verdad? A lo que alguien se anima y hace alguna pregunta, yo anoté aquí algunas things to ask. A pesar de que comentaste que no hubo ningún major shift en, en microbial composition, sí notamos ciertas leves diferencias en abundancia entre los diferentes organismos. Em, so, ¿Tú crees que debido, aunque son cambios pequeños, tú crees que estos cambios pequeños pueden ser algún factor que esté predisponiendo a los sea urchins de hoy día a que padezcan de la enfermedad causada por el ciliado?
Al revés. Yo creo que el ciliado tiene una comunidad microbiana que se asocia a la enfermedad. Yo no creo que hay suficientes diferencias en el erizo que tiene enfermedad y el que no, como para decir que la enfermedad está causando un cambio drástico. Pero sí creo que esta bacteria, al solamente estar asociada con los erizos abnormales, erizos que tienen la enfermedad, creo que sí él puede estar asociado al ciliado en sí.
Ok, perfecto. ¿Alguien tiene alguna pregunta del público? ¿No? ¿Verdad? Comentaste que una de las comparaciones que hiciste que fue entre el Caribe y África. Eh, ¿Puedes abundar un poco más sobre eso? ¿Verdad? ¿Cuál es tu hipótesis de cómo es posible, ¿verdad? Que a pesar de que están tan distantes y obviamente hay factores ambientales que contribuyen a, a ese ecosistema en donde está viviendo el erizo, ¿cómo es posible, ¿verdad? Que allá esté la misma composición que acá en el Caribe.
Pues me encanta esa pregunta. La composición microbiana es bien distinta en cuanto al erizo, porque el erizo que vimos en África es Equinotrex diadema, no es diadema antilarum. Así que la composición microbiana es completamente distinta, pero la única bacteria que se mantiene constante es esta bacteria asociada a la enfermedad. Por eso es que nos llama mucho la atención. Encima de eso, lo que vemos es que, por eso es que pienso, y la tesis es que la bacteria está asociada al ciliado, no necesariamente bacteria ambiental que viene debido a la enfermedad. Porque si la bacteria está asociada al ciliado, pues se pueden mover en sintonía y pueden moverse a la misma vez. Puede ser un endosimbionte, puede ser simbiótico. Si
Exacto.
está dentro del ciliado o no, aún no se sabe, pero mi hipótesis es que la bacteria es un endosimbionte del ciliado y al nosotros estar viendo las muestras como composición, no podemos diferenciar dónde está la bacteria, si es del erizo o del ciliado en sí.
Exacto. Yo creo que una de las cosas que encuentro más interesante de todo es cómo dentro de un mismo ecosistema tienes un erizo enfermo y justo al lado tienes uno sano. Entonces, ¿cómo es eso, verdad? Eso también de nuevo, ¿cómo es eso achievable? Porque básicamente están compartiendo el mismo ecosistema, básicamente se están alimentando de lo mismo, y uno está enfermo y el otro no.
Por eso es que tenemos muestras y por eso es que si vemos otra vez la gráfica de taxonomía, se ve que la bacteria Fungia onconensis está presente en algunos de nuestros erizos normales que están en el área de enfermedad. Nosotros entendemos que debido a haber colectado en cierto día, el erizo no estaba enfermo en ese momento, nosotros lo clasificamos como no estar enfermo, pero sí puede ser que tenga el organismo y es que no haya tenido enfermedad visible aún.
Ujú. Sí, a lo mejor lo cogiste en el early stage, en el que todavía no tenía ningún tipo de síntomas, pero iba en camino a.
Demuestra la importancia de tener métodos de detección como lo es el PCR para el ciliado o las bacterias asociadas.
Exacto. Em, por el momento no veo que tengamos ninguna otra pregunta, así que ¿verdad? Te extiendo las gracias por haber estado en el día de hoy, habernos compartido toda esta información sobre el erizo negro. Y nada, le hago la invitación a todos los que están conectados en el día de hoy a que llenen el formulario de evaluación y también que se conecten entonces para la próxima charla que tenemos la semana que viene, que es el martes, que es por el doctor Lester Rosario, que nos va a estar hablando sobre tips de propuestas para la hora de de someter tanto a nivel graduado como luego a nivel postdoctoral. Así que muchas gracias a todos y nos vemos en la próxima.